Hello, everyone. Thank you for joining. Good afternoon. My name is Oля Videnskaya. I am going to talk about the lipidomics and lipids in neuroscience today. The talk that we're gonna go through today is basically built on based on three blocks. First, we'll go through the introduction. Then, and basically during the introduction, we'll talk about the neurons and glia cells. We will then go through the processes that are happening in our bodies normally. These are both myelination and remyelination. And then we'll talk very briefly about the applications of mass spec to particular diseases. Today we'll cover Parkinson's disease, Alzheimer's disease, and multiple sclerosis. Now, in this introduction, we will discuss the main brain components, basically neurons and glia cells. We will have a look at both human and murine brains and their specific lipid compositions. We'll be exploring neurons and glial lipidomics, and this will allow us to set a foundation for studying the specific roles of lipids in health and in different neurological conditions. So when we started, we thought, let's check the lipid profiles of different types of central nervous system cells. And of course, none of this work is we would be able to do single-handedly. We had a wonderful partner, German Center for Neurodegenerative Diseases, and many of the examples I'm going to talk to you today about were performed together with the DZED and EAM. So the central nervous system and peripheral nervous system consist of electrically excitable cells, neurons, and the cells that do not produce electrical impulses, but they provide vital support for the whole system. They're called glia cells. And glia cells in the CNS include many different types of cells, such as oligodendrocytes, astrocytes, and microglia. The primary function of oligodendrocyte is to generate myelin. Astrocytes, they recycle neurotransmitters, they shape synaptic circuits and maintain the blood barrier transfer. And microglia are mainly related to immune responses and brain homeostasis and play a crucial role in myelination and remyelinations. And those are the types of cells that were studied in this particular work. As key component of neural cells, lipids hold significant information in the efforts to better understand the human brain and its diseases. And of course, detailed molecular lipid analysis is an adequate tool for the cell type resolving assessment of lipid composition and lipid metabolism. About 75% of mammalian cells lipid species are exclusively found in neural tissues. So lipids contribute to extreme morphological and functional diversity of the central nervous system that consists, as I mentioned, of neurons and glia cells. Here you can see the molecular composition of human brain and the average distribution of major molecules in the human brain. And if we recalculate it to the dry weight, then lipids account for more than 50% of the human dry weight of the brain. And so specific lipids and lipid classes and sometimes even lipid species are known to be enriched in specific cell types. And this is something that we were going to focus today on. But one example I think that is known for all of you is that neurons and myelin contain high levels of cholesterols, then galactosyl ceramides, for example, are typically accumulating in the membranes of oligodendrocytes during myelation. So all these things and specific lipids for particular processes and applications, this is something that we're going to talk about today. Another thing that we think is important to underline is that many neurological and neurolipidomics research is happening on mouse brain, and anatomically speaking, mouse brain serves a powerful animal model to study the brain and the human brain in its diseases, be it mental or physical disorders. The genes responsible for building and operating both mouse and human brain are highly concordant, and in principle, the cellular architecture of both human and mouse brain tissues is well conserved, and they're organized. It's organized in a very similar fashion and contain a comparable diversity of cell types. So this is why, in many cases, we're going to refer to a mouse brain. Now, together with DK and SET, we aim to generate a cell type resolved lipidomic profile of the mouse brain in order to acquire the basic data set, the fundamental data set for further neurobiological research. And researchers applied lipidomics analysis to differentiate the lipid compositions of neurons and of different glia cell types, including already mentioned astrocytes, oligodendrocytes, and microglia. Overall, 23 lipid classes were analyzed, quantified, and they comprised around 750 individual lipid species. Out of 23, in 13 lipid classes comprised more than 99% of all quantified lipids. Phosphatidylcholine and cholesterol were the most abundant, most common lipid classes in all 4 sample types. And phosphatidylcholine and yes, they were the most abundant. And then when comparing for the different cell types, we of course saw some particular differences in the lipidomic profiles in both lipid classes and lipid species. Here you can see the PCA plot, a principal component analysis that was used to visualize variation and highlight robust patterns in the data sets in a 2-dimensional manner, basically, score plot. And so here we see that the different cell types, for example, oligodendrocytes, they cluster together, and then microglia clusters together, neurons clusters together. So basically we see that they have distinct lipid profiles. We also see that oligodendrocytes were the most divergent cell types, and probably this is due to their role in the myelin production that, of course, comprises the unique lipid composition. And if we do the deep dive into the lipid composition itself, then we see that in neurons, for example, there are phosphatidylcholine, phosphatidylethanolamin, and cholesterol are the most abundant species. And in principle, though otherwise not rich in phosphatidylglycerols, lipid species data show that they were also enriched in selected phosphatidylglycerol lipids. Oligodendrocytes were most divergent cell types, as we saw in the previous graph already, and again, most likely due to their myelin production, they have a distinct lipid composition. So they have sulfatide and hexacylceramides, they're enriched, and those are both lipid classes that are structural lipids of myelin. Interestingly, microglia were rich in phosphatidylglycerols and sphingomyelins, and it contained a high level of specific sphingomyelin lipids that were almost absent in neurons and oligodendrocytes. And finally, astrocytes contained higher level of 2 types of phospholipids, PS and PI, so phosphatidylserine and phosphatidylinositol, and also diacylglycerols. Though otherwise it was not enriched in phosphatidylethanolamines, they were also particularly rich in 2 specific lipid species of phosphatidylethanolamine. Overall, we can say that each neural cell type features a distinct lipid composition, thus expanding in the research project and to the question of whether each cell types adapts specific lipid metabolism pathway too. So basically we saw that lipid accounts for over 50% of human dry weight, and we do see distinct lipid composition of each cell type. Then this was also published. You can check this paper online. And now we can move on to the processes having this background. So basically, what we want to see was the ganglioside lipidomics in CNS developmental myelination. And for this, we needed to develop a particular method that was also done together with DZ and E. And if you check the gangliosides that play a huge role in myelination, we see that it's actually not so easy. So ganglioside structures, they're very specific. They contain galactose, sialic acid, and some combinations of those. They're more hydrophilic, and they're super difficult to extract and measure. So therefore, we needed to come up with a particular extraction method from mice samples, and in this case, several steps extraction procedure was used to make sure that the material amount of only 1.5 micrograms is sufficient. So we used water phase, and we used organic phase, and then used reverse phase solid phase extraction, and performed further than the shotgun lipidomics. And then we were allowed using this method to see the alteration in ganglioside content within the murine brain. So this particular lipid species we saw during the changes we saw were during the early myelination, then nearly complete myelination, so 12 and 21 postnatal days, and then to the adult stage. And we do indeed see that the highest quantities, for example, were observed in GD1 species, followed by GT1. Here in this case, we see it, and lower amounts are found for then GM1, and basically we were able to see this ganglioside content changes throughout the developmental stages. And the highest quantities were observed, as I already mentioned, in GD1 species, followed by GT1. And then basically we were able to see that the most abundant ganglioside measures were in GT1 3612 and GT1 3612, as well as the further changes within this lipidome. So and we also saw some increases and some decreases throughout this whole myelination stage. So this was basically it. We did develop the method allowing to measure ganglioside lipidome in the CNS tissues and cells. We analyzed also, I didn't really have a chance to show it, but we also analyzed the mice LAG genes, that LAG genes synthesizing most gangliosides. And we also saw that the mutation caused near total absence of brain gangliosides. Interestingly, the compatibility of this shotgun method with a broader global lipidomic profiling techniques, it does represent a significant technical advancement, and that allowed us and our partners and collaborators further look into this area of research. Again, this was published a couple of years ago, so you can check if you want this paper fully. The next one is the process that we want to have a look at is the remyelination. And in this particular case, we worked together with the Technical University of Munich, and we wanted to check what is the role of lipid metabolism and lipid droplets in the process of remyelination. Remyelination overall it's a regenerative process that can occur during central nervous system damage. And further, we'll talk about it in the context of Alzheimer's disease, for example, multiple sclerosis. And so in this case, remyelination unfortunately often fails during the progressive phases of a disease. So we wanted to look deeper into mechanistical processes that are happening during the remyelination. So this is a bit of a closer look into the myelin injury. As we already discussed today, microglia and innate immune cells of the brain and spinal cord, and they are essential player in inflammation response and myelin repair after demyelinating injury. So in case of CNS injury, myelin debris, so-called myelin debris, is generated from the myelin sheath covering axon. And here you can see major lipid components of myelin debris, such as cholesterol, ceramides, sphingolipids, and phospholipids. And under normal conditions, this debris is being cleared. But the biggest part of the biggest obstacle is to deal with cholesterol. And in principle, again, under normal conditions, this cholesterol debris, unlike other lipids, cannot be degraded by the lysosomal enzymes and is transferred from late endosomes to endoplasmic reticulum. And ER on its turn cannot really cope with the excessive amount of free cholesterol. That is why free cholesterol is being esterified into cholesterol esters and is stored in lipid droplets. And so basically this cholesterol exposure overall is required in microglia macrophages to start this response of the myelin debris uptake. And this buffering mechanism of a lipid droplet sometimes can get damaged, for example, in the presence of TREM2 mutation. And these were the model that was analyzed in this case. So in case of TREM2 mutation, we did see the number of lipid droplets that are needed for proper resolving of myelin injury dropped in case of TREM2 knockout. And that we also saw that the lipidomic analysis actually reveals that TREM2 deficient phagocytes not only fail to generate cholesterol esters, but also are deficient in other lipid species like TAGs. And the accumulation of phosphatidic acid in TREM2 deficient phagocytes points to the decreased activity of particular phosphatases, which catalyze the conversion of phosphatidic acid to diacylglycerols. We also saw that the evidence of the ER stress, this of course was not done by mass spec, but I think but probably by Western blot. I'd have to double check, but in principle, in these cells, we also saw the evidence of the increased ER stress. So basically, we performed the study that investigated the phagocyte function of clearing cholesterol excess that occurs as a result of demyelinating injury. And in summary, cholesterol efflux and esterification are essential to promote proper resolution of this immune inflammation and proper remyelination. And if this doesn't happen, then unfortunately ER stress kicks in the cell and then in the cells, and then we see the consequences of the ER stress. So to sum up, remyelination requires myelin debris clearance, and then microglia phagocytic function relies on the lipid droplet genesis, and TREM2 receptor is essential for lipid droplet formation. This was also published. You can check this article if you want. And now, having all this knowledge at hand, we can finally move to the applications to the diseases. I think we will start with the multiple sclerosis. Multiple sclerosis is one of the most well-known neurodegenerative diseases. It's a chronic inflammatory disease also of a neurosystem, and basically it manifests in the body, attacking the myelin sheath of neurons in both brain and spinal cord. And the damage causes people some communication problems between CNS and the rest of the body. Globally, around 3 million people live with the multiple sclerosis, and of course symptoms may vary drastically. Yeah, so this is a very important part of neurolipidomics to study multiple sclerosis and other neurodegenerative diseases to make sure that we do our part of trying to help patients and research in this field. Now, in this particular study, again performed together with DID-CNA, we wanted to check what are the lipid biomarkers for multiple sclerosis. And again, this is the most commonly used article, the image during this presentation. So basically, in terms of underlying pathological processes, researchers noticed that there are changes in lipid metabolism in patients, and lipid analysis, that there is a hope that this lipid analysis can discover specific biomarkers for multiple sclerosis to support fast and accurate diagnosis and maybe better treatment and monitoring of interventions and prognosis. So basically, what was done, it was the twin study. And interestingly enough, the result of the twin study revealed no major difference in total lipid content between the twin sibling with the MS and without. However, interestingly enough, on the molecular level, there were two species, two lipid classes basically, that were involved in, that were basically seeing some differences that were reduced in the twins with the multiple sclerosis. So in particular, 44% of ether-linked PCO and PE on 47 of PEO lipid species were altered. Interestingly enough, the top hits were for PCO and PCO minus, and it contained polyunsaturated fatty acids. For example, here you see the two lipid species that were interestingly involved in the patients with multiple sclerosis. So this is PC and PCO minus, both with polyunsaturated fatty acids. I think in this particular case, it's arachidonic acid. And then this was interesting. The reduction of ether lipids and polyunsaturated fatty acids in patients with MS indicated altered lipid signaling in multiple sclerosis. And among the 73 twin pairs in the cohort, and especially those with MS, 20 patients didn't receive any treatment for multiple sclerosis, and still the altered lipidome profile remained independent on whether there was a treatment or not. So this is something that clearly is not linked to MS treatment, but to the disease itself. So multiple sclerosis, just to sum up, damages the lipid-rich myelin nerve cells. Lipid profile alterations are hallmark of multiple sclerosis, and lipidomics help to identify possible lipid biomarkers for diagnosis and maybe potentially one day for treatment monitoring. Again, you can read this paper online. Now I think we can move to another article, another case study that was performed on Alzheimer's disease. Yeah, we see that there is basically it's another progressive neurodegeneration. So during Alzheimer's disease, there is a damage to neurons and also loss of communication between neurons. And like many other actually conditions and diseases, it is linked to aging. And unfortunately, the actual cause, it is currently unknown. And a lot of people struggle with it, and there is a need to understand the pathological, molecular, and signaling pathways that are involved in Alzheimer's disease development. What we wanted to do is to check if multi-omics analysis can, of course, including lipidomics as our part, can reveal molecular pathways in Alzheimer's disease to improve the predictive power. This was done together with Nestle Health Science Institute, and basically here you can see that The other omics analysis were used, so proteomics, carbon metabolism, metabolomics, and neuroinflammation markers were checked. In this study, patients with normal cognition, with mild cognitive impairment, and mild dementia were worked with. The cerebral spinal fluid samples were collected, and then our part was to perform lipidomics, and then we built together with other omics some prediction models for Alzheimer and prediction for cognitive decline. We don't have much time, unfortunately, to go through the details, so I'll just have to briefly scroll through a couple of next slides. Basically, the multiomics factor analysis allowed us to see that there is a particular molecule, CE16-0, the cholesterol ester, was associated with the Alzheimer's disease and might be used as a potential biomarker for the disease, at least for some particular subset of these patients. And so we do see that Alzheimer's disease metabolism is complex. However, of course, multi-omic studies do help to check the molecular pathology. And new molecular pathways in Alzheimer's can explain some variants and predict disease progression. If you're interested in more depth analysis, you can check this particular study that was done and published. And then final part of today's presentation, just a couple more minutes, is Parkinson's disease. In this particular study was performed together with Harvard Medical School, and we wanted to check with our collaborators what is the role of lipases in the Parkinson's disease development. Overall, again, it's more than 10,000 people, 10 million people living with Parkinson's disease. So this is really, really essential for public health care that these things are taken care of. Now, Parkinson's disease and other nucleopathies cause an accumulation of protein that is called alpha-synuclein, and it gets misfolded, and then it is formed into Lewy bodies and neurites, and they cluster and they cluster in the brain cells, and this basically is a hallmark of Parkinson's disease. So what our colleagues wanted to check is that there's the particular role of lipases in neurolipid synthesis and degradation in the Parkinson's disease development, and they wanted to test a potential new therapeutic target, triglycerol lipase called LIPE, or LIPE we call it internally, that break down tags. And basically they did a knockdown, they did a pharmacological inhibition in cell culture and in primary cells from patients with Parkinson's, and they checked for the fatty acid composition, and we do see that the knockdown, lipid knockdown, decreases monosaturated fatty acids, and in both cases, actually, for the knockdown and pharmacological inhibition. And so we saw similar effects of pharmacological inhibition on the primary neurons, and basically all these studies also allowed us to check the not only triglycerols, but also phospholipid species. And we do see a large drop with the lipid inhibition in PS, PC, PI, and PE phospholipid species containing 16-1 fatty acid. And yeah, basically those were the things that primarily are related to lipid part of this study. So we do see that lipid affects the fatty acid pool, and targeting fatty acid generation via lipid rescues Parkinson's disease phenotype. This has been also published. If you want, you can check these publications. Actually, I'll be happy to. So if you can find this article, but you can just drop me a line and or drop us all a line through a contact form, and we'll help you to navigate through this. Basically, that's all I wanted to tell you. Unfortunately, we're pretty limited with time, but once again, we went through the types of neurons and glia cells, through myelination and remyelination, and through Parkinson's disease, Alzheimer's disease, and multiple sclerosis. Also, if you want, you can take our survey to get our neuroscience lipidomics ebook. Just scan the QR code, and you can read in much better detail all these cases I talked to you about today, because we only are limited to, like, showing you a couple of slides. They're proper things you can find in this lipidomics ebook. And I think now my half an hour is almost over, so I will be very happy to then move on to the questions and answers. So I'll be happy to hear your questions and maybe provide you with the answers. Please, let's start the discussion.